we understand the mechanism of transcription and translation, let us see what is the machinery required in the cell for protein synthesis. It's a very elaborate process, but happens within a very short period of time. It requires the following components. Following components are involved. Following components are involved. 1. DNA. DNA provides the code. DNA provides the code for each of the protein required by the cell. So if a cell requires 3000 types of protein, that cell should have 3000 types of codes. 3 million types of proteins, 3 million types of codes. So DNA provides the code for each of the protein required by the cell. mRNA. mRNA copies the message in the code by what we have described as transcription, copies the message in the code and converts them into a sequence of codons, sequence of triplet codons. These codons should contain the initiator codon, the sensible codons and the terminator codon. tRNA molecule is meant for identifying the amino acid, acids involved in protein synthesis with the help of specific anticodons. Anticodons are complementary to the codons in the mRNA. So identifying the amino acids with the help of specific anticodons and transporting them to the ribosomes. Then, now this particular situation, identifying the amino acids, picking up the amino acids and transporting them to the ribosomes, this is called adaptation. Each tRNA molecule adopts a particular amino acid based on its anticodon. Suppose, for example, methionine is to be transported to the ribosomes. Methionine is coded by AUG. So AUG is the codon for methionine. This methionine molecule can be picked up, identified and picked up by a tRNA molecule, only that tRNA molecule which has a complementary arrangement of bases in its anticodon U, A, C. A ಗೆ U for U, A for G, C. This is called anticodon. So only this tRNA molecule, a tRNA molecule having UAC as the anticodon, only that can carry methionine. Then ribosomal RNA. Ribosomal RNA. Ribosomal RNA for arranging the amino acids brought by tRNA into a polypeptide on what basis? Based on the sequence of codons in mRNA. Then ribosomes. Ribosomes are the actual sites of synthesis of proteins. Remember, that is why they are called protein factories of the cell. So it is in the ribosomes that rRNA is involved in what is called as translation. Then amino acids. The raw materials for protein synthesis. Raw materials for protein synthesis. ATP molecules. For activating the amino acids. ATP molecules bind with the amino acids and activate the amino acids so that these activated amino acids can be picked up by the tRNA molecules. So this is called, this process is called activation. The entire mechanism requires a series of enzymes. DNA helicase for unwinding the DNA, RNA polymerases for attaching the RNA nucleotides during the synthesis of mRNA, DNA ligase for rejoining the helices of DNA, and so on. A variety of enzymes within RNA polymerase. If you take, for example, eukaryotes, there are 3 types of RNA polymerases: RNA polymerase 1, RNA polymerase 2, RNA polymerase 3, and so on. Then certain divalent metallic ions, like for example magnesium ions, manganese ions, are necessary for the enzymes to function. So these are the components in the cell that are actually involved in protein synthesis. This entire set of components forms the machinery required for protein synthesis. It means essentially transcription, translation, activation, and adaptation. Transcription, adaptation, activation, and transcription. These are the essential steps in protein synthesis. Transcription is a step that occurs in the nucleus in eukaryotic cells, and translation is a step that occurs in cytoplasm. But in a prokaryotic cell, it is only in the cytoplasm that both transcription and translation take place. mRNA synthesized during transcription comes out of the nucleus, reaches the cytoplasm, and immediately begins translation. When transcription is in progress in the nucleus, in the cytoplasm, activation of amino acids by ATP molecules, and adaptation, identification and picking up of the activated amino acids by tRNA molecules will be completed, so that the tRNA molecules can involve themselves in translation as soon as mRNA reaches the cytoplasm.